My name's Jonathan Napier and I am a flagship leader here at Rothamsted Research, and I lead our GM program on making omega-3 fish oils in plants, but I'm also in charge of a working group that is looking at both gene editing and GM and how that can be incorporated into our future programs. So, we're really interested in trying to enhance the nutritional composition of plants. We've got several GE projects in my team looking at trying to use gene editing to improve the crop we're interested in, which is camelina, which is like an oilseed crop that's related to oilseed rape canola. And we use gene editing to try and knock out, to block a particular pathway so that the plant doesn't make something that we don't want it to make. So we're basically using gene editing as a fine-tuning mechanism to improve the oil content of our camelina plant. So what we discovered with the research we've been doing is that gene editing is really a very powerful tool to precisely change the composition of a plant. And so we used it to specifically inhibit a particular gene, and that then changed the oil profile in our camelina plants. And we did it with two different examples, one where we changed it so that the oil actually became more like you'd find in olive oil, and another example we changed it so it didn't have any erucic acid, which is sometimes found in crops like oilseed rape. So we realized that it's a really powerful technique and it's a very precise technique. I think GE has amazing potential. I think it can really revolutionize plant breeding because it can do things to a degree of precision and speed that we couldn't previously do with any other forms of plant breeding. So therefore I think it's going to really transform the way that agriculture is done, in the same way as gene editing is transforming all aspects of life sciences and the biomedical sciences. It's completely changing the way that we think about how we go about doing things. I think one real potential area that you could use gene editing for, which would be really powerful and deliver real societal benefit, would be using gene editing to remove allergens and toxic proteins that naturally occur in plants and foodstuffs. So, for example, what happened, if you could remove allergenic proteins from sesame or peanut, you could make a foodstuff that would then not be toxic, lethal to some people in society. In the same way as you could use gene editing to remove the allergenic proteins from wheat that cause celiac disease. That would be a great thing to do. Or you could use gene editing to remove ricin, which is like the second most toxic protein on the planet. You could remove ricin from castor bean. There's lots of things you could use gene editing for that would deliver real and immediate benefit to society. And I think it would be a shame not to use it.